mine. I am a medical technologist at Akron Children's in the molecular diagnostics department. And then also my other time, I am also a PhD candidate in biomedical sciences at Kent State as well. So just a little bit about me. And so today, I will be discussing with you all DNA polymorphisms in human identification, which I will pull up my PowerPoint. Let's see if I can get it.
Malia, did you make any changes to the PowerPoint?
Yes, I do have a couple, some slight changes.
Okay. If you want, at some point today after the lecture, if you want to send it to me so I can upload it for the students so they have the most current one.
Absolutely.
Thank you.
So are you all able to see my screen?
Not yet.
No, not right
Not yet.
There it is. We see it now.
Okay, great. Thank you. So at any point in time during the lecture, please let me know if you have any questions or if I'm going too fast. Feel free to stop me. Just say, hey, Malia, like slow down. Or if you want me to explain something, please let me know. Okay, so as I stated earlier, I will be talking about DNA polymorphisms and human identification purposes under molecular diagnostics. So you're probably wondering what on earth is a polymorphism? So when we talk about polymorphisms, DNA polymorphisms is really just a slight sequence difference when looking at a reference standard that's kind of present in about 1 to 2% of the population. So only a few individuals might have this slight sequence difference. Polymorphisms is what really makes us unique. And because the human genome is so large, there are so many areas and possibilities to have these slight changes in our DNA sequences. These changes can be single bases or it could be thousands of bases at a time. Not all of them usually have a phenotypic effect. For instance, they may not cause a disease or anything like that. It's just a change in the nucleotide sequence and nothing really occurs there. And so, like I said, they're throughout the whole entire genome. And if we know the location of these variable areas on the genome, we can use it as landmarks for locating genes or specific regions. And with these polymorphic markers that are designated throughout the genome, there are typically different alleles, or another way of saying that is different versions. A lot of them are inherited, meaning you get one version from your mom and one version from your dad, and you have your own unique combination, which is why it's so important and so easy to use this concept for human identification purposes. And so there are 4 main types of polymorphism. I will only be discussing 3 out of the 4 today. For the first one is restriction fragment length polymorphism. The second is variable number tandem repeats. Now, VNTRs are not so commonly used due to the fact that they're so large and it would take weeks to actually process these type of samples, so we don't typically use VNTRs. But STRs, short tandem repeats, are very common because they're very, very short, really easy to amplify through PCR and easy to visualize as well on a gel. And then lastly, there are single nucleotide polymorphisms as well. So we will be discussing RFLP, STRs, and SNPs today. So starting with restriction fragment length polymorphisms, essentially fragment lengths basically vary depending on the location of these recognition sites. And so we have different enzymes that will make cuts along the DNA at specific sites. And depending on if you have a polymorphism or a slight change in your DNA sequence, it can end up changing the amount of cuts that you have on your DNA. And therefore, we can separate those fragments in order to have some sort of identification for that particular person. Now, obviously with RFLP, it is going to be largely dependent on the location of your restriction sites in reference to a particular gene of interest that you may be looking at or anything to that specific question. So for here, if you take a look at the top, we have normal DNA, and let's say that ECO-R1 site, that's a specific enzyme recognition site. This restriction fragment enzyme will actually come and make a cut at the DNA at the specific site. And let's say that, for instance, you have a point mutation, meaning you have a change in base pairs. And this could happen, and because of this, essentially the site won't get recognized, and therefore you won't have a cut along that site. You could also have insertion of DNA at that particular site where it might actually make multiple cuts. You could also have duplications, which will also result in another cut along that DNA fragment. And then you could also have a whole fragment insertion or deletions, which will ultimately vary the amount of fragments that your DNA will have after it has been digested with this enzyme. And so to kind of get a better visual of this, remember, RFLP is from changes in the actual fragment size, okay? And so let's say we take BGL2, and this is a recognition site. And so the enzyme, when you add it to the DNA, it's going to look for these specific sites and make a cut. And so let's say that we know that there are 2 recognition sites that's most commonly found in the population. And let's say for this first line, person number 1 is positive for the first cut site and second cut site. So if you make 2 cuts along this fragment, you should ultimately end up with 3 full fragments, which we will actually see here in this gel. And then for person number 2, they do not have the second recognition site, and therefore they only get 1 cut, which would result in 2 fragments. So their genotype is positive or plus and minus for these recognition sites. Person number 3, we see here that they do not have the first recognition site, but they have the second recognition site. It still results in 2 fragments here, but you'll notice here that there's a slight difference in the size of the fragments. So we're able to still distinguish where the cuts are actually made along the genome. And then here, they don't have any of the recognition sites, so therefore no cuts will be made. And if no cuts are made, then you'll just have one long band, which is why it's sitting here at the top. And so when you're using RFLP, you're really looking at the pattern of fragments that are produced based off the number of restriction sites that are located in your genome. And so typically we'll use gel electrophoresis or capillary electrophoresis, which I'll talk a little bit more about capillary electrophoresis a little bit later. Okay. So RFLPs, the genotypes are inherited. Like I said, most of these polymorphisms that we'll talk about today are inherited. So you, at each locus or each location on your genome, you get 1 allele from one parent and 1 allele from the other. So for instance, let's say this is our DNA ladder on our gel. And at locus A, you'll get 1 of these alleles from your father, and you get 1 of these alleles from your mother. And you can see here as the child profile, you'll see that each of these alleles had came from each of a parent at each of the locus as well. And so because of that, you're able to actually do some parental testing using RFLP. And of course, I'm definitely going to make you guys do a little bit of an activity as well. So take a few minutes and actually go through this scenario. So which alleged father's genotype has the paternal alleles? So this is your child and this is the mother. Between these 2 options, which one would be the father of this child? I'll give you about 2 minutes to look at it, but if someone wants to, turn their mic on and have a guess, that would be great.
Would it be the AF2?
Nice. Awesome job. Absolutely. So AF2 would be the father of this child. And if some of you did not quite understand how that works, you know that child A has these two fragments here at locus A. You know that based off the purple coloring from the mother, that they had to have gotten this allele from mother A. And in order to have this one here, they still could have gotten it from either AF1 or AF2. So then we move to the next locus, locus B. Locus B, we know they got this allele from their mother. And in order to determine which father had this allele, obviously the father had to have had the allele in order to pass it. And we see here that AF1 did not have that allele at all. So there's no way that this allele could have come from father 1. So therefore, AF2 would be our father. And you see here how between the two fathers, some of them actually have similar alleles. And in order to make more of these paternity tests more accurate, we always add multiple locus for a lot of these paternal tests in order to increase our confidence in, for surely saying, okay, this is the father of this child. And there's no percentage that would actually say that it could possibly be someone else. Okay. So next, I'm going to skip down to our STRs. So short tandem repeats are just essentially a repeat of a particular nucleotide sequence. STRs only range from 1 to about 8 or 9 nucleotides at a time. You could have a mononucleotide, which is just the same base pair repeated over and over again. You could have a dinucleotide, which is 2 nucleotides that are constantly repeated, like AT, AT, AT. And then you could also have a trinucleotide sequence, which you would have 3 nucleotides constantly repeated. And so different alleles are going to have a different number of repeats. And so here's the distinguishing factor between RFLP and STR. So with STR, you're going to have different chunks of those repeats at a time. And each allele is also inherited from your parents as well. So if we're looking at these two examples, they are the same grouping of allele nucleotide sequences, so TTC. But here you notice that there are a different number of repeats. And so because of those different number of repeats, you are ultimately going to have a different length in these alleles overall, which makes it easy for us to separate through gel electrophoresis or capillary electrophoresis. And so a further example of this, STRs, like I said, can be analyzed based off the amplicon size. So if you know you have a specific repeat region, which we do designate certain ones with certain names. So for instance, this is TH01. It's designated with TCAT, and you'll see this repeated constantly. And so for here at the top, we notice that there are 7 repeats of this particular loci on the genome. And for allele 2, which is also going to be the TH01, and you'll notice here that it has a different amount of repeats. Now, if you remember earlier, I said that STRs can also be or are also inherited from your parents. So allele 1 is going to come from mom or dad, and allele 2 is also going to come from mom or dad, which is why you'll get some variability on the amount of repeats. And so anytime you run a gel electrophoresis, we always have to have allelic ladders that are our standards that represent what is common in that population for that particular loci. And so when you run a gel electrophoresis, you'll notice that there's 2 ladders on each side. And for each individual lane is an individual person. And so for here, for allele 1, this is our first person here. They have 7 repeats at allele 1 and 8 repeats of allele 2. So therefore, you're going to see 2 separate bands, okay? And this is an example of another person. Here, they also have a allele for 7 and then they have allele for 10. So they also have two different bands. Does someone know what this lane would look like if this person only had 2 7 repeats? How would this lane look if there were 2 7 repeats? Would you still see 2 fragments? Anyone want to give me an answer?
I'll take a guess. Would you only have one because they're both the same size or whatever?
Yes, excellent. Thank you so much. So yes, you would only have one band. So if you're homozygous for a particular allele, you're only going to get one band because the bands won't separate. They'll all move together. So that's the only difference you'll see if you have a homozygous at a particular spot. So nice job. Okay. Okay, so from this previous image, this is what would it look like if you did gel, if you just did a gel electrophoresis. Now, like I said, even before that, when I was talking about how we like to add a whole bunch of loci so that we can for surely identify a particular child to their parent or anything like that. Well, because of that, there also is a technique to analyze STRs looking at multiplex PCR. So multiplex PCR is when you use multiple primers in order to amplify multiple locations on the genome so that we can actually look at the genotypes across different areas across the genome at the same time. Now, the biggest limiting factor about doing multiplex PCR is that the allelic ladders that you run cannot overlap in the same reaction. So if you actually look at these particular areas designated on the genome, you'll notice that the allelic ladders actually do not overlap each other. And that is really important because they need to have some sort of separation so that when we call a lot of the STRs and, be able to identify the actual genotypes at these particular loci, we want to make sure that the allelic ladder is easily visible for those particular loci. So a little bit of the limiting factors with doing multiplex PCR. Now, the other way to analyze STRs, most of the examples I gave you thus far have been by gel electrophoresis. You have your individual allelic ladder per loci, and then you have your patient samples in between. The other technique is to do capillary electrophoresis, and your final results actually turn out something a little bit like this report here. And so essentially your alleles here are going to be compared to an allelic ladder as well, but that ladder gets run through simultaneously at the same time with your amplicons here, whereas it's in its own separate lane in gel electrophoresis. So it might be a little hard to kind of imagine, which is why I have a nice little overview of capillary electrophoresis and how that works. And so let's say we're looking at one loci in a patient. At allele 1, they have 5 repeats, and at allele 2, they have 11 repeats. So essentially, you would shoot this DNA through the capillary electrophoresis here. This usually has a polymer in which your DNA is going to travel through. And does anyone know the charge of DNA?
Is it positively charged?
It's actually negatively charged. So because of the phosphate backbone of DNA, it actually has an overall negative charge. And because it has that negative charge, when you run an electrical current through this capillary, it usually goes from negative to positive. And because this other end is positive, that negatively charged DNA is going to want to travel as quickly as possible to get to that other side. Now, because DNA has an overall negative charge and you put electrical current through the gel electrophoresis, ultimately, what's going to separate your DNA fragments is the size. So essentially, the larger fragments are going to take a longer time to get to the other side versus the shorter fragments, which are going to move really quick through that polymer that's going to be in this capillary. Each of your fragments have a fluorescent tag to them. So by the time they get to, I call it like the finish line, or they get to the laser, that laser is going to make them fluoresce, and the detector is going to keep track of that particular fragment. And because there is a standard that's run through the gel electrophoresis, it also corresponds to the base pair, the number of base pairs that each of those fragments are, okay? And so, for instance, when you put these two alleles, or the DNA fragment as a whole, through the capillary electrophoresis, we know that the 5 repeat is going to come first because it's shorter. And here it corresponds to this ladder, and we know the amount of base pairs looks like it's about 55 base pairs in total, which corresponds to 5 repeats. Here, the detector actually is able to calculate each of those base pairs, and as far as like how many repeats that actually correlates to, which is really nice. And then the 11 repeats will come and it gets fluoresced, and corresponding to the standard markers, it is a little bit larger, and we know that that is going to be 11. So at this particular loci, we know that this person has the genotype 511. So how do we use STRs when we are doing like paternity testing or anything of that nature? So like I said, genotypes are inherited. You get one allele from one parent and an allele from another parent. And so, for instance, let's say that we have 5 loci, or 5 locations that we're specifically looking at. And the amylogenin, which I'll talk about in a little bit, is actually really specific to Y chromosome. So if you're a male, this would be something that you would have. But let's say that we have a child, which is our C at the top here, and we're looking at these specific loci. And then also we have our mother's profile here at these specific areas as well, and our father's here as well. So you'll see here that from the child, it's a mixture of your mom and your dad's alleles here. So we know that this peak had to have come from mom because it's not present in the father. And we know that this peak had to have come from the father because it's not coming from the mother, okay? And the concept is very similar as well to the RFLP, in that if you do not have, if you're homozygous for a particular allele, you only really have one peak, okay? So you'll see here mother and father also had the same allele here, and therefore, or I'm sorry, the father here only has one peak, which would mean that he was homozygous for this particular loci, okay? So he only could give the child one of these, and then from the other, you could see he got it from the mom. Does this make sense as far as the STR loci and how they're inherited from mother and father, and how you could get kind of a mixture profile, but you could use those to go against your mother and father to see where you're getting your alleles from? Just give me a little yes or no.
Makes sense to me.
Okay, thank you. All right. So amylogenin, specifically that locus, it's not a short tandem repeat. That's really important to note. But it is specific to the Y chromosome, and it's a lot bigger than the X, than it is on the X chromosome. So essentially, anytime you're seeing amylogenin on a profile, that usually is what we use to determine sex, okay? And so here, if you have a mixture, an X and a Y, we know that this is a male. And as a female, if you only have X, you're only going to get one peak here, and that is going to be our female. Okay. So a little bit more about the Y chromosome. Your Y chromosome only comes from your father. There's not any recombination that happens. And so any STRs that are Y chromosome specific are things that you're looking for your paternal lineage. Okay, so it's a haplotype because it only comes from one parent. And so, like I said, it's usually used for exclusion analysis if you're just looking more at your paternal lineage versus your maternal lineage. And so any paternally related males are going to have the same YSTR profile. And so you're probably like, well, yeah, okay, that's great. You can do paternal lineage, but what about your maternal lineage? Well, I'm so glad you asked. Because for maternal lineage, we actually use mitochondrial DNA polymorphisms. So in case you did not know, you actually have two types of genomes within your cell. So we all know that the mitochondria is the powerhouse of the cell, but did you know that the mitochondria has its own DNA? That DNA is actually specifically circular, so we're actually easily able to isolate, especially in like research purposes. But on the mitochondrial DNA, there are two hypervariable regions, and those that are closely related to each other are going to have similar sequences versus those that aren't as related are not going to really have the same sort of variation at these two particular regions. And so we can use mitochondrial DNA polymorphisms as a way to exclude individuals, or if we're just looking really at the maternal lineage. So the Y chromosome is going to be related to the paternal lineage, and then you have the mitochondrial DNA, which is going to be related to the maternal lineage. So those are very unique in those aspects. So, of course, quick activity, make sure everyone's good. Which alleged father has the child's paternal alleles? Okay, so here you have the child's alleles at these particular loci, and then you also have the mother and her particular alleles at these different locus. So if you could figure out if it's going to be one or two. So I'll give you a couple, like 2 minutes or so, and whoever wants to say it out loud, go right for it.
Is it father one?
Nice. Get that little thing to come up. Nice job. Yes, father number one. And so essentially, here you'll probably notice like, oh, okay, these both have 17 here, so how can you distinguish? Well, that's why, like I said, it's always important to include multiple locus on a test so that we can discriminate easier based off that fact. So they both have 17, so they both could have given it to the child here. We know that they had to have gotten 16 from the mother. If we move on down to another locus here, father 1 has 14, 15, father 2 has 16, 17. We have 16, 18 from the mother. So the child had to have gotten 18 from the mother. So we're trying to figure out where they got allele 14 from. And automatically looking at the second line here, we can see that they could have only gotten 14 from father number 1. For scientists, even though, like, yeah, theoretically you could be like, yeah, I could stop right there. Typically, it's good science to always add as many locus as possible, because probability says that you can multiply these chances and say, hey, it's 1 to the 1,000th chance that it could be someone else that happens to have the same profile. So if those of you that are like slightly interested in forensics, typically when a scientist has to sit on the bench, and a lot of the prosecutors are like, well, how could you say it's my client? Technically, can't someone else have the same allelic profile as my client? And typically, scientists usually always have to state the facts and also have to state the statistics. And typically, because there's so many loci that are actually on a lot of these profiles, most of the time, scientists will say the chances of having the same allelic profile as, the person that's getting prosecuted would be, if there were theoretically 3 Earths, it would have to be another person on the 3rd Earth. So because of like really wild statistics like that, a lot of science, when it comes to doing a lot of these profiles, are pretty bulletproof, I'd say, for the most part. So that's why a lot of these profiles will have multiple loci in order to increase the significance of the statistics for this. So getting more into the clinical aspect of using STRs would be engraftment testing. And so specifically for allogenic bone marrow transplants, just so we're all on the same page, allogenic is when you have a mixture of cells. It's all from the same species, but you can tell it's 2 different individuals. And so essentially, this is essentially meaning receiving a bone marrow transplant. And so we use engraftment testing to see how effective a bone marrow transplant is, specifically for cancer or diseases of the immune system. And so essentially, these transplants can be used actually monitoring STRs, which is really cool. And so essentially, when a recipient receives a bone marrow transplant, they are going to have their cells are going to show a chimeric profile, meaning they'll have cells from their own body and also a mixture of cells from their donor as well. And so to kind of look at it a little bit more of a case study, that kind of gives you a better idea of how this works in application. Typically, before a transplant occurs, usually a doctor needs to order a STR analysis of the donor and then also a STR analysis of the patient themselves in order to see what are the loci that are shared, or I'm sorry, what are the alleles at each loci that are shared between the donor and the patient, and which ones that are different between the patient and the donor. Okay, and typically when you get these STR profiles in real life, you'll notice that you'll see these little, they're called stutter peaks. And typically, it's just an error from PCR. Typically, the whole entire fragment didn't get amplified, and so that's why you get these tiny little peaks here and there. That's completely normal. Typically, there's always a threshold value when you are analyzing these peaks. And so essentially, it might be a little difficult to tell here, but the first line here is either the recipient or donor. The second line is also recipient or donor, and so on and so forth. So you can see here where they may differ, and you can see here where they actually might share a peak. So you're probably wondering, okay, that's great. You can see what's the same and what's the difference. Well, how is this really important as far as monitoring bone marrow transplant patients? And so I looked through the Journal of Molecular Diagnostics to actually show you guys how this really looks in real life. So for this specific case, this was a 48-year-old woman with chronic myeloid leukemia, and she had received an allogenic peripheral blood stem cell transplant from a male donor. And so essentially, as I stated earlier, usually you need a STR profile of the donor and the recipient to see what's the same and what's the different. So we know that the donor here has 2 different alleles at this particular loci. And going to this loci here, we know that the donor recipient share they share this 15 allele, but they do not share the second allele here at this particular loci. And so so after the recipient receives the donor transplant here, another STR profile is usually done to follow up to see how well or how well the recipient is with the donor's cell transplant here. So if we look at line C here, this is 3 months after the transplant was done. And you'll notice here that the profile of the recipient is actually identical to its donor, which is really good. This means that the patient is in remission. And then another follow-up was made 6 months, and here's where things get a little bit dicey. So here you notice that there actually is a mixture here. So the recipient's alleles are starting to essentially make a comeback in mixture with the donor. And so this is sort of the look of remission. Excuse me, not remission, but a failure of the donor's stem cell transplant. So in a molecular diagnostics lab, specifically our lab, we actually do engraftments and we do the test, the genetic testing in order to get these STR profiles. We actually call each of these peaks, and then that report gets sent to the doctor where they will actually do the clinical interpretation of that. So that's something that we actually do here at Akron Children's, which is pretty cool stuff. Any questions on engraftment testing? Okay, so I will keep it moving. And lastly, I will talk about S&P haplotypes. So S&Ps are inherited as well, just as everything else we've talked about today. It can be used a lot for gene mapping, human identification, chimerism analysis, and other different applications. And SNPs are really important, especially for some of the ongoing projects that have started in the 90s. But specifically, there is the Human Haplotype Mapping Project, which actually is going through and surveying all the people across the world to see what are common SNPs throughout the human genome, which is really cool. And then I have this brief 2-minute video on SNPs that's really cute, that kind of makes it really easy to understand, that I will share with you all.
The farmer's dog. They make and deliver fresh and healthy human-grade dog food right to your door. But here's the thing.
An entire set of 23 human chromosomes is called a genome. The human genome is composed of 3 billion base pairs. Variation at a single base pair is called a SNP, or single nucleotide polymorphism. When the body makes new cells, it doesn't make many mistakes. But nobody's perfect. Sometimes, when the genome is copied to make a new cell, a single base pair gets left out, added, or substituted. Single base pair substitutions create SNPs. There are around 10 million SNPs in the human genome, which account for many of the genetic differences between you and everyone else on the planet. Some SNPs account for differences in appearance. Others can affect how we develop diseases or respond to drugs. Most SNPs, however, seem to lead to no observable differences between people at all. Since variants are passed down from one generation to the next, the number of differences between your DNA and your neighbors can tell you how closely you are related to each other.
And so another thing to also that's really important from that video is that most SNPs are not disease causing, but there are a small handful that do actually cause diseases. Most commonly, have you all heard of sickle cell anemia?
Yes.
Yes.
So sickle cell is actually caused by a single nucleotide polymorphism, specifically in the first exon of the beta globulin gene. And because of that, it causes that sickle shape, unfortunately, and that is how you get sickle cell. So there are lots of applications for SNP, especially if you're looking for a cause of a disease. Like I said, very few diseases are caused by one SNP per se, but a lot of genome sequencing can actually detect for those SNPs compared to this database that has been established in order to make a diagnosis for a lot of patients. So. And to that, are there any questions on the lecture today?